our next session is the lightning talks. So these were submitted abstracts that we selected from in order to give quick 5-minute discussions. There will not be time for questions, but you know, if people have questions, they can post it, and perhaps the panelists can kind of type in answers offline. So the first panelist that we have, or the first lightning talk that we have today, is from Anne Yoder at Duke University.
Thanks, Jeff, and sorry for the, you know, if I jumped the gun on sharing my screen. So I want to thank Circulomics and Arima for the invitation. This is really exciting, and the talks so far have been just super. So I'm going to talk to you about field genomics and biodiversity documentation in Madagascar. In case you don't know, Madagascar is one of the Earth's hottest biodiversity hotspots, with 80 to 90% species endemism across all groups. And all of it is severely threatened by human impacts. So it is a hotspot indeed. So part of this consequence is that species populations and their genomes are vanishing before our very eyes. And I am particularly interested in all 3 levels, and I'm using genomics to examine the problem. Okay, so we have, as a group, many goals, but here are some of our primary goals. We want to document the biodiversity, and we're using, presently we're using a DNA barcoding approach for that. We are wanting to scale up to population genomics with whole genome resequencing approaches, and very importantly, we want to transfer this knowledge to the Malagasy scientific and academic community and the conservation community. The example I'm going to use is my favorite group of organisms, the mouse lemurs of Madagascar. And so these are, you're looking at 6 different species here, but as you will see, they pretty much look the same. So these are cryptic species. They're genetically highly diverged, but morphologically and ecologically cryptic. So to even know which animal you're looking at, we are using a DNA barcoding approach with cytochrome B, and then we use that in the context of a phylogeny just to find out which lineage we are looking at. So this is an important aspect of our field work. And in the before times, or you know, in the longer sense, this could take months to years to accomplish, so that, you know, the samples would be collected in the field, and then we'd have to go through all of the permitting issues with U.S. Fish and Wildlife Service, and then finally get the samples back to the lab and you know, do our genomics work at that point. But we have transitioned into a field genomics approach, and that the first publication from that effort was published this summer in Conservation Genetics. So we're using Oxford Nanopore and mini PCR technologies and taking those to the field and doing the barcoding on site and finding out instantaneously which animals we're looking at. So one of our field sites that we're particularly interested in is in the southeast region of Madagascar. And this was identified back in 2009 and 2011 using microsatellite data as a hybrid zone, so that there was putative gene flow among mouse lemur lineages in this region. Now, if you look at this photograph, it looks pretty homogeneous, but if you look very closely, you'll see that the near this little stream, it's more of a gallery forest type of habitat, and it's much drier away from the stream. And what my colleague, Jor Ganshorn, who's been working there for many years, says is that you only see Griseo rufus, which is a dry specialist in the dry forest, and Murinus, which is more of a generalist in the gallery forest. They thought they were seeing hybrids between these 2 species. Using these microsatellite data, we have now applied RAD-seq approaches to these very same samples and found that there are no hybrids, at least not in this data set. So we're working on this, and this is a paper that's about to come out on the bioRxiv, and you can look at what the problems are with the microsatellite approach. So going forward, what we want to do is, as I said, take a population genomics approach using whole genome resequencing, but we need to start with really excellent reference genomes. We have one at this point that was published in 2017. We kind of threw the book at it using Illumina, PacBio, Hi-C, also optical mapping with BioNano. And it, so it's a pretty nice genome. And amazingly, we were actually able to characterize the centromeres because very randomly and accidentally, one of the Nick enzymes falls within one of the monomers of the centromere. And Karen, I noticed that Beth Sullivan was one of your peeps in your group, and she was our collaborator on this project. So she verified the centromeres with using a fish approach. So I speak to both Circulomics and Arima and ask you to help me figure out how we can do telomere to telomere sequencing for small samples from small animals collected under challenging field conditions. And in fact, what brought me to this symposium was learning about DNA guard. So I think that's going to be a very important tool going forward. And I really look forward to talking to everyone at both companies about this. So just to wrap up, a lot of our work is facilitated by a new lab that's been built in Antananarivo, which is the capital city in Madagascar, Mahaliana, and the founders are collaborators on this project. And three of my co-authors on this paper, Lantu here, Lydia and Marina, have held several workshops to demonstrate the power of the mobile lab, again, just for DNA barcoding. And it's been quite popular, and there's a lot of excitement in the Malagasy academic community for incorporating genomics into their field projects. So with that, I will thank my collaborators and co-authors on this paper, Marina, Lydia, Fidi, Liz, Rachel, Lantu, and Peter, and to our funders. And I think that is it for me. Thank you very much.
Thank you, Anne. That was really interesting, and those lemurs are super cute.
The curse of the cute, I call it.
Yeah. All right. So actually, I forgot to mention at the beginning that these the four panelists here or the four Lightning Talk panelists are vying for a genome to be constructed by Circulomics and Arima. So good luck to all of the Lightning Talk panelists. So up next we have Ida Heyer from Uppsala University.
Hi everyone, I am Ida Heyer. I'm a PhD candidate from SciLife Lab at Uppsala University. So just quickly, first want to thank Arima and Circulomics for giving me this opportunity to present some of my work here today. So I named my talk Amplification-free long-read sequencing for detection of Cas9 off-target activity. I think most of you are familiar with the CRISPR-Cas9 system and how it's a very popular tool for genome editing. But also when we talk about CRISPR-Cas9, we have to think about the off-target effects that they can cause. And it's typically, an off-target effect occurs when the CRISPR-Cas9 complex sits down somewhere in the genome where you did not intend it to sit down, and it makes a digestion of the DNA at that spot and introduces mutations. And these off targets can be predicted. It's typically done by sequence comparison. There are also some experimental tools to do this, and they usually rely on short read sequencing. So what we have done, we have developed two different protocols to utilize long read sequencing for detection of Cas9 off targets. And the first one, we use PacBio, and we call it Smart OTS. And OTS is for off target sequencing. And the other one is for Oxford Nanopore, and it's called Nano OTS. I'm not going to go into detail exactly how these protocol works, but I just want to mention that for both of them, we use random fragmentation of the genomic DNA. And then later in the protocol, we cut these molecules with the Cas9 enzyme. And when you have sequenced these molecules and mapped them to the reference genome, you end up with a really specific read pattern, as you can see here, where all of the reads basically start or end at the same position. And this is how we can detect both the on-target and the off-target sites. And to evaluate our methods, we designed guide RNAs for 13 different targets. And we detected over 100 Cas9 cleavage sites for all of these guide RNAs. And this is just an example for one of the guide RNAs targeting the ATXN10 gene. So on the left here, you have the on-target site for both of the technologies. So here you can see the Cas9 cleavage site and this really typical read pattern that we get. And on the right here, we have an example of one of the off targets. And again, you see this same read pattern as we see for the on target. And if you compare the genomic sequence at this off target with the guide RNA, they are very similar. And for this particular off target, we have a 3 base pair mismatch between the guide RNA and the genomic sequence. And this table just summarizes all the off targets that we found for this particular guide RNA that you have on the top, that sequence, and our on target doesn't have any mismatches to that, but then you have all the off targets listed below. And for this particular one, we found 5 base pair mismatch as the biggest difference. And to really emphasize why we think long read sequencing is a good approach for detecting Cas9 off targets. To demonstrate this, we designed the guide RNAs that towards the so-called dark spots in the genome. And in this example, we designed a guide RNA against the STRC gene. And when you sequence this gene with the Illumina short reads, you get some spots that doesn't get any coverage at all. And this is also something that Everdnall showed in a paper from 2019. So our guide is designed towards one of these dark regions, but that doesn't affect the outcome for our sequencing, and we get this really this read pattern that we saw before, that we can clearly see the Cas9 cleavage site, and we also have coverage over the entire locus. So this is something that we think is really beneficial with using these methods. And if you are a bit more interested in what I've been talking about, we have a preprint out at bioRxiv that you can go ahead and check out. And with that, I just quickly want to thank everybody involved in this project, and especially my co-supervisor Adam Amar, who did a lot of work on this project as well. And of course, Arima, Circulomics, and all the attendees for listening. So thank you.
All right, thank you very much, Ida. Up next, we have Mikko Rautiainen from NIH NHGRI.
Okay, hi everyone. So first some background. So the Bruijn graphs have been used for purposes such as genome assembly, read error correction, and variant calling. And the Bruijn graphs are built from k-mers, which are substrings of length k. Here on the right, you can see an example. So the input sequence is on the top, and in this example, k is equal to 3. So all substrings of length 3 are extracted as k-mers, which you can see here. And then the k-mers are the nodes in the Bruijn graph. So the Bruijn graphs have traditionally been built from short Illumina reads. The reason for this is that the Bruijn graphs require a low read error rate. So CLR and ONT reads are usually not used with the Bruijn graphs. But now that we have hi-fi reads, which have long read lengths and low error rates, we can again use the Bruijn graphs based methods. So for this reason, we implemented MBG, which is a tool for building sparse Bruijn graphs designed specifically for hi-fi reads. And we used MBG as a part of the telomere to telomere project to assemble some of the most difficult repeat regions in the human genome. So in a sparse Bruijn graph, instead of using all k-mers, we only use a subset of the k-mers. You can see an example here on the right. Here's the input sequence. And instead of using all k-mers, we might pick, for example, just these k-mers. Now, because the number of k-mers is smaller, we can build sparse Bruijn graphs much faster than regular Bruijn graphs. And we use minimizer windowing to select the k-mers, which is where the name comes from, M for minimizer and BG for the Bruijn graph. So we did some experiments with MBG. First, we compared MBG to bcom2, which is a previous tool for building the Bruijn graphs. We used both tools to build graphs of E. coli from hi-fi reads. And I'm not going to go through these numbers, but the conclusion is that MBG is an order of magnitude faster and uses much less memory than previous methods. The reason for this is the sparse k-mer selection shown in the last slide. And in fact, MBG can assemble E. coli into a single contig in 8 seconds. And the second experiment is that we built whole human genome graphs of HG002 using hi-fi reads. And the conclusion here is that MBG can build whole human genome graphs in just a few hours on a single CPU core. Finally, for more details, you can see our preprint on bioRxiv, and the code for MBG is on GitHub, and MBG can also be installed with Bioconda. Thank you.
All right, thank you. That was really interesting. On to the next lightning talk. This is Brandon Pickett from Brigham Young University. All right. Brandon, do you see? I'm not seeing any slides here.
I saw it for a second.
Yeah.
Sorry, I had it for a second and then I forgot to include the part to share sound, and now my PowerPoint disappeared on me. Huh. I'm sorry about that.
I can share your PowerPoint if you want. I have it
Okay,
up.
I found it again. Sorry, I'm
Okay,
not
you got
sure
it?
what
Okay.
happened, but there we go.
Okay.
Oh, and that's the end of it too. Gosh, I'm on top of it. Okay, my name is Brandon, and I just want to share briefly some work we've been doing to assemble and annotate a genome for the giant trevally, which is a marine apex predator. I just want to show you one of the benefits of doing non-model organisms. We get some cool pictures. So this is a picture of the one that we sequenced, caught off the coast of Molokai in Hawaii. Giant trevallies can be over 5 feet long and up to 120 pounds. They're a really popular sport fish, often found throughout the Indo-Pacific. And this one in particular was caught about half a mile off the coast of Molokai, just outside the reef. It's not letting me advance. Here we go. I want to also show you this video of the fish being landed. It's short, but it's just kind of fun to see. Okay, so some justification for why we're doing this work. First, population declines generally in large-bodied reef-associated fish predators has been declining. You can see on the graph on the right-hand side, the iluas, or these trevallies, the populations have been declining, often from overfishing and habitat loss. understandably, a lot of the money and research that's gone towards fish like this, such as tuna and swordfish, goes because of commercial reasons. Even though these are really popular sport fish, the giant trevallies, they don't have near the resources put at them. And any work that we've done, well, I guess I haven't done, but work that has been done in the past has been focused on single or just a couple genes with fairly low resolution to get at population structure and whether there's hybridization between other species of trevallies. So for this study that we've been working on, we did an analysis more on a genome scale, and we had 225 samples throughout the Indo-Pacific where we were able to do dd-rad sequencing. It allowed us access to over 32,000 SNPs for our analysis. So for the genome assembly itself, so far we've generated PacBio continuous long reads data with about 84x coverage. We were able to do Illumina RNA sequencing on 8 different tissues, which we're hopeful to use for annotation. And then we've also ordered Hi-C, and we had an issue in our lab. So we had to reorder it, and it's currently on the machine now. So I'm excited to get that going. As far as the assembly statistics so far, which we used Canoe for, so shout out to Sergey Karen, who's also a participant today. We have about a 600 megabase genome, and we were able to just straight out of Canoe with no extra filtering or anything get 7.5 megabase N50, NG50, and an area under the N curve of about 9 million. And so we were pretty happy with that to start with, and we're excited to see where it goes from here. So as far as the results from this and the next steps, certainly with the genome, we have Hi-C planned in the works, and we plan to annotate and release that data. More on the population genomics side, the analysis has revealed that even though these fish are large-bodied and capable of traveling across the Indo-Pacific barrier, they don't. We're not entirely sure why, but that's what the evidence reveals. And we've also seen population structure in Hawaii, and so our initial recommendations, because of this work, has been to put precautionary management measures in place until we can really get at what's going on. Then, of course, we'll be able to move forward and do that in future work. But that's where it's at so far, and I appreciate your time listening. I just want to acknowledge my advisors and collaborators, Perry and Keone, and then Jessica, who works at the South African Institute for Aquatic Diversity, who's helped with the population genomics side of this analysis. If you have any questions, I'd love to hear from you outside of this, because we don't have questions.